let me continue on that recording. And I am one of the PIs of the 6 PIs that make up the genetic research centers of the NIDDK Inflammatory Bowel Disease Genetics Consortium in the United States and Canada. And this is our second in a series of webinars on inflammatory bowel disease and its relationship to genetics and issues in our challenges in genetics of IBD. And today we're very honored to have Dr. Jonathan Pritchard from Stanford University join us. Dermot McGovern, my colleague at Cedars-Sinai in Los Angeles, will be introducing Dr. Pritchard. And I'm just going to have a few words why we're interested so much in Jonathan speaking with us today. So as many of you know, we have now over 240 loci in inflammatory bowel disease for European populations, but for non-European populations, probably less than a quarter of those loci to 10% of those loci have been truly defined. And we're seeing different effects on the different loci in different populations. And when we try to use new tools like the genetic risk scores, we're finding that they don't apply from one population to another. And Dr. Pritchard's done work that's showing within populations you have to account for age and various effects to use these polygenic risk scores to help make predictions. Also, we're struggling with all the non-coding regions and how they translate to genetic risk, as well as differences in RNA expression. And Dr. Pritchard also has great expertise in these areas. So we're really looking forward to his talk today that's going to, and the work that he's done overall in genetic variation and in populations that will help guide us. And now Dermot will introduce Dr. Pritchard.
Thanks, Steve. Yeah, it's a great pleasure to introduce Jonathan Pritchard. Actually, he needs little introduction to those of us interested in complex disease genetics. Nevertheless, I'm going to do my best in 30 seconds or so. He's currently the Bing Professor of Population Studies at the Department of Biology and Genetics at Stanford University. Previously a Howard Hughes investigator, as well as Professor of Human Genetics in Chicago, where he crossed paths with Judy Cho, I was just hearing about. Prior to his primary degree, I also just found out that he did a lot of his schooling in the UK, and we were just exchanging some stories about Oxford. His primary degree was in biology and what I would call maths or math. I don't know what you call it, Jonathan, at Penn State before completing his PhD at Stanford. And then a postdoc with Peter Donnelly in Oxford, where we briefly overlapped, though we don't knowingly remember meeting each other. His CV is littered with prizes, including the Kistler Prize in Population Genetics and the Edward Nowitzki Prize from the Genetic Society of America. His group has authored many papers. I note that one has been cited more than 26,000 times, which is pretty incredible. His lab studies genetic variation and evolution and genotype and phenotype relationships, and also spends a lot of time thinking about why does so much of the genome contribute to the genetic basis of complex traits. So, Jonathan, we are delighted to have you here and look forward to your talk about why human complex traits are enormously polygenic, lessons from molecular biomarker traits. Thanks very much for your time.
Okay. Well, Dermot, thank you so much for the introduction, Steve, and also to Judy for the invitation to speak here today. Okay. Can you see this okay? Great. All right, good. Yeah, so it's really great to get to talk to you today, and thanks very much for the invitation, and I'm looking forward to the discussion in the second half. So one thing that we've been interested in for several years in the lab is the question of why we get so many hits in GWAS. And so I'll explain in a minute what I mean by that question. Before I go on, I just want to highlight a few of the key people who've been in my lab over the last few years, shown at the bottom here, who've driven a bunch of these projects. In particular, Nas and Sahin have led the work on the biomarkers that I'll be talking about, as well as Evan and Young, who started these projects about 4 or 5 years ago. So historically, if we think about our intuition in genetics, a lot of this has come from studies of simple and single gene traits, even going back to the 19th century with Mendel's work. Of course, one of the huge strokes of luck or genius that Mendel had was that he stumbled across these traits that followed simple Mendelian inheritance patterns. Of course, in humans, there's actually very few common traits that follow simple Mendelian inheritance patterns. Some of the traits that do, of course, are Mendelian disorders. So in the 1980s and the 1990s, there started to be enormous progress in dissecting genetics of Mendelian disorders, where there are individual mutations that have that are destroying the function or changing the function of a gene in such a way that it's got a really reproducible and predictable effect on a trait. And of course, these kinds of one gene to disease or one gene to trait kinds of examples have been really helpful for our understanding of how genetics works, but it's really not typical. Most phenotypic variation is highly complex, as we all know here in human genetics now. If you look around a room or think about all the people on the Zoom call, most of the ways that we differ one from another are enormously complex in the sense that they are driven by very large numbers of genetic variants. And so I think that this raises a fundamental question that is, why does so much of the genome affect any given trait? So how should we think about the fact that there are perhaps 10,000 variants or more that affect height or disease risk for some disease, and that these variants are spread very broadly across the genome? What does that tell us about the mapping from genetic variation to phenotype? It seems very different from the scenario of a mutation in a single gene that causes a disease in a relatively deterministic way. So I'm going to start out with a couple of examples just to give you a sense of how I think about these problems. I think that schizophrenia is a relatively extreme example of this. So in the latest published meta-analysis, which is about 6 years old now, they had 108 genome-wide significant loci. And so, of course, for that's using about 200,000 individuals. And of course, that's quite an achievement to identify so many significant loci for a really difficult disease to study. And yeah, of course, one of the interesting findings from schizophrenia, as well as from many other traits, has been that the lead SNPs are only responsible for a small fraction of the expected heritability. So about 10% in this case. Meanwhile, there's an enormous amount of additional signal that's lurking down here in the bottom of the Manhattan plot, and all of the SNP variation together is estimated actually to explain about 80% of the heritability. So the vast majority of the heritability is coming from this enormous number of small effect loci that are spread relatively broadly across the genome. The other point I would make is that for schizophrenia, and I think it's fair to say for most disease traits, the vast majority of the lead hits are difficult to interpret. And I think actually in IBD, I'm of course not an expert in IBD, but my sense is that actually you have the great fortune that a number of the lead hits are actually much more interpretable. So for example, in schizophrenia, with 108 loci, really there were no gene sets that were significantly enriched at all. The lead gene sets, the lead gene sets are actually associated with synaptic function and brain function. So even though they're not significant, you might imagine actually that they are onto something that's biologically real. But my point is that for schizophrenia, as well as many other complex traits, the lead hits that you get out tend to be very, very difficult to interpret. The other point I want to make is that schizophrenia and height, which is another trait that we've studied as kind of a model trait, have enormous numbers of variants. So we've estimated perhaps on the order of 100,000 variants that affect both height and schizophrenia across the genome. And so then the question is, if we think of a scenario where you have 10,000, 100,000 variants spread very broadly, what's the nature of the mapping from genotype to phenotype? How does this work? And so we've thought about this in the context of a number of different traits, but what I'm going to tell you about today is some work that we've done on two traits that we think are somewhat simpler, that we regard as kind of model systems. And these are urate and testosterone. So I'm going to spend the next few minutes telling you about GWAS that we've done on UK Biobank data for both of these traits, and how these illuminate the biology of these two traits, as well as the overall genetic architecture. And I think that these can be instructive for thinking about other complex traits as well. So we became interested in urate and testosterone and other molecular traits because we think, number one, because they are molecular, they sort of sit somewhere in between the complexity of, on the one extreme, organism level phenotypes like disease or height or BMI, and on the other end, cellular level traits like for example, EQTLs. Secondly, we actually know a great deal about the secondly, we actually know a great deal about the biology of urate and testosterone in advance, and so that actually makes the GWAS data much more interpretable. I should say about urate that it's a small molecule that's a byproduct, produced as a metabolic byproduct of the purine synthesis pathway. The levels in the bloodstream are tightly regulated. When you have excess urate, it actually crystallizes out to form these sharp needles in the joints, which cause gout. There's a great deal of data in the UK Biobank for urate. They've measured urate as well as about 30 other metabolites in 430,000 individuals. And what I'm going to be talking about today is the GWAS for urate in the quote-unquote white British subset of the UK Biobank. And I should say that all of this work has been led by two people in my lab, NASA Sinnott Armstrong, who's an amazingly talented graduate student, and Sahin Naqvi, who's a postdoc who's also done wonderful work on this. So, as I mentioned a moment ago, unlike schizophrenia or perhaps probably most other diseases, we know a great deal in advance about the processes that might regulate urate. And so, for example, we know in great detail what the urate synthesis pathway is. So this is shown here, the biochemical synthesis pathway. And everywhere that there's an arrow on this pathway, we actually know what the enzymes are that are catalyzing these transitions. So these are obvious candidate genes that we might expect could potentially be involved in urate levels. Secondly, a lot of the control of urate comes from kidney function. So the kidneys are involved in basically exporting urate out of the bloodstream into the urine, as well as reuptaking urate from the urine back into the blood to maintain appropriate urate levels in the bloodstream. And we actually know from prior literature what all of the transport genes are. So again, these are natural candidate genes that we might think of as in advance as being involved in urate levels. Okay, so now let's go to looking at the GWAS data in about 330,000 individuals in the UK Biobank. And here it is. So the lead hits marked in green are all involved in transport of urate in the kidney. So every peak that is marked with green text here contains one of the urate solute carrier genes that we had annotated from previous literature. So the second category of genes that we see a bunch of hits for are transcription factors that are involved in liver and kidney development, shown down here in blue. Lastly, among the top hits, there are a couple of other genes that are marked in red and whose function is a little bit less clear, but they're involved in, they're also hits for a bunch of other biomarkers. So they're clearly doing something important, though we don't really understand their biology at this point. Now, although the top hits are really highly interpretable, one thing that's striking to us is that there's actually more than 200 independent genome-wide significant signals. So there's an enormous number of different loci across the genome that are contributing to this trait, even though it might seem like a relatively simpler trait. And in a few minutes, I'll show you that actually we think that the true number of causal variants affecting urate is probably more on the order of 10,000 total variants across the genome. Okay, so if we put these hits back onto the this plot of candidate genes that I showed you from before, you can see in green are all of the genes where we have a genome-wide significant hit within 50 kb, and you can see that actually 8 out of the 10 genes that we had annotated have a genome-wide significant hit nearby. And this is about a 37-fold enrichment relative to the genome-wide background. So this is really huge enrichment for this pathway here. Secondly, we do see enrichment in the biosynthesis pathway. So all of the gene names that are marked in red also have genome-wide significant hits nearby. In gray, indicate numbers of other genes that are annotated here that don't have hits. And so here there is a significant enrichment. It's about 2-fold, but it's really not dramatic in the same way that it is for the transporter genes. So we kind of get the sense here that the production of urate is having a relatively minor impact on the levels in the blood, but actually what is really mainly controlling urate levels are the import and export of urate inside the kidneys. So the last point that I want to make on this is that despite the really big hits that we see and how concentrated they are in and around these transporter genes and kidney TFs, most of the heritability is actually driven by a large number of SNPs that sit in kidney regulatory chromatin. So in this regard, what we see for urate is like a lot of other complex traits where we see enrichment for signal in active regulatory regions spread broadly across the genome. So this is one analysis of this type where I'm showing you the fold enrichment for SNP heritability in active chromatin regions in different types of tissues. So, so starting with kidney up at the top here, there's about a 37-fold enrichment per SNP for heritability for SNPs that are inside active chromatin in kidneys versus outside. And it turns out that actually this explains a large fraction of the signal. So if we actually remove kidney active chromatin from the model and then look at other tissues, then none of the other tissues are clearly show much substantial enrichment. So perhaps adrenal and pancreas is showing some significant enrichment here, but it's relatively modest. Same for liver. It's relatively modest compared to what's going on in kidney. But the main point I want to make is that even though for kidney we see very strong enrichment around these critical genes, actually most of the heritability is coming from this large polygenic background in active kidney chromatin. The next example that I want to show you, and the second example for the model traits, is in circulating levels of testosterone. And here I think the story has got some similarities and some interesting differences. So of course, testosterone has anabolic roles in building building muscle, for example, in both sexes. It's responsible for secondary sex characteristics in males. It's at much higher levels in males, so about 10-fold higher average testosterone levels in males versus females. Again, we know a great deal about the the processes that control testosterone. So for example, here I'm showing the chemical biosynthesis pathway that produces testosterone. Up at the top is cholesterol, and so this is so there's steroid biosynthetic pathway. There's also a bunch of other familiar genes on this, so cortisol here, estradiol, and other estrogen-related molecules here. And so there's a bunch of important molecules that come out of this, this pathway. And again, everywhere there's an arrow here, we actually know what the key genes are that catalyze those steps. Okay, so now we just, based on some preliminary analysis, we decided to run the testosterone GWAS separately in males and females. When we look at this in males, what we see, when we look at this in females, what we see is that there's clear enrichment of hits that are in these biosynthesis genes marked in green, and this is a large and highly significant enrichment. We also see some we also see a small number of hits that are in genes involved in gonad development in females. In the GWAS, the Manhattan plot for males actually looks quite different. So there's almost no overlap between the hits that we get in males versus females. And the biggest hits are involved in control of a protein called SHBG, which is steroid hormone binding globulin, and this is actually a molecule that that binds testosterone and controls its and makes it bioinactive. And I'll talk about this in a minute, but it turns out that SHBG induces a negative feedback loop. Induces a feedback loop so that when you have higher levels of SHBG, it actually winds up stimulating testosterone production, but it only does that in males. And this helps to explain why we see this strong SHBG component in males, but not in females. So I kind of hinted at this looking at the Manhattan plots, that the peaks are not overlapping. And in fact, interestingly enough, that the male and female testosterone male and female testosterone turn out to be sort of essentially unrelated traits. So one way to look at this is to take hits that are genome-wide significant in either males or females and plot the effect sizes in one sex against the other. And you can see here that there's no correlation at all. And when we do this genome-wide using LD score regression, we find that the genetic correlation between males and females again is 0. So this was really very striking to us. Even though we see sex differences in these two traits, we do know that there are important aspects of the biology that are shared. So, for example, they're both producing testosterone out of the same biosynthetic pathway, and it just seemed counterintuitive to me that we would see no overlap at all. So we decided to dig into this a bit more, and what we find is that actually the processes that are regulating testosterone in males and females wind up being quite different, and so the GWAS is very illuminating for this purpose. So first of all, steroid synthesis turns out to be important for both males and females, but in really different parts of the pathway. So here's the biosynthetic pathway that I showed you before, but what I'm doing now is labeling the genes that have hits in according to whether they have hits in males or in females. So the genes that are marked in purple have hits in males, the genes that are marked in orange have hits in females, and then the genes in black have hits in both. And you can see there's just a couple of those. And so you kind of get a sense here that that the male hits are in this part of the pathway that are sort of clustered close to testosterone, whereas the female hits tend to be in the upper part of the pathway. And so we think that actually there's kind of some kind of trade-off between the part of the pathway that sort of is producing aldosterone and cortisol and other related molecules over here versus flowing in to produce testosterone down here. And so in females, where much more of the testosterone is produced in the adrenal gland, which produces a lot of the aldosterone and cortisol, this is actually controlling this actually has a substantial, the genes that are active in this upper part of the pathway have a substantial impact on testosterone production, whereas in males, most of the testosterone is produced in the testis, and so this part of the pathway winds up being less relevant. Beyond that, it turns out that there are very important differences in regulation of testosterone levels, as well as I mentioned the main tissues of production. So in males, there's this important feedback loop that's heavily regulated through the HPG axis. So this is the hypothalamus pituitary to gonadal axis. And so basically you have positive positive regulation that's coming from the hypothalamus and pituitary. It's flowing through luteinizing hormone, and that's stimulating testosterone production in the testis. You also have this negative feedback loop, so that when circulating levels of testosterone in the blood are high, then that produces negative feedback. So for example, for sports players like Barry Bonds who take excess testosterone, this actually has a dampening effect on the hypothalamus and pituitary, which leads to reduction in testis size. Now, the importance of SHBG is that it actually takes some of the testosterone out of circulation, so it becomes bioinactive, and that reduces the negative feedback loop. So you get basically a negative correlation between the amount of SHBG and the amount of testosterone that is produced. So in females, in principle, the same pathway exists, but actually the positive effects from luteinizing hormone to testosterone production is much more complicated because there are multiple products that are stimulated by a luteinizing hormone, and also the overall amount of negative feedback is very, very low just because there's not enough circulating testosterone to really drive that feedback. And so what we find is that actually the hits so what we find in males is that we actually get a large number of hits that are all involved in this positive part of the feedback loop that's shown in purple. So every gene here marked in purple is a genome-wide significant hit in males. That's sort of in this upper part of the hypothalamus and pituitary pathway. We also see positive genetic correlations between luteinizing hormone and testosterone production. And I'm not showing it here, but again, we see these strong signals from SHBG. And in contrast, none of these genes are hits in females. So we think that together, that helps to explain these sex differences. But the next point that I want to make on this is that although these molecular traits highlight these core pathways very clearly, they're still extraordinarily polygenic. And so this is a plot that we made to where we split the genome up into roughly megabase bins, and then we estimated SNP heritability in each bin across the genome from chromosome 1 to 22, and so the x-axis here shows you the cumulative amount of heritability adding across all of these bins, and then the bins that contain these important core genes are marked in orange. And what you can see is that although these core processes show clear enrichment, and they're highly significantly enriched, they're still only contributing a relatively modest fraction of the heritability. So in the case of testosterone, they're contributing together, they're contributing about 10% of the heritability through these core processes in both males and females, and most of the background is coming from a huge number of variants spread across the genome. And I'm not going to get into the details, but we've estimated that there's on the order of around 10,000 variants that affect each of these traits that we've looked at. So this is a lot of variants, like spread quite widely across the genome, although we'll note it is quite a lot less than we get for height, where we estimate it's about 80,000. So what I've shown you so far is that for a wide variety of traits and diseases, we see the heritability is spread very widely across the genome. We see that genes with trait-relevant functions contribute only a small fraction of the total disease risk, although the biggest hits are often enriched in relevant gene sets. And lastly, that the variants that contribute are highly concentrated in regions that are active chromatin in the relevant tissues. And we take this to imply that most effects are flowing through effects on gene regulation. So this raises an important question that I want to talk about in the last few minutes, which is, how should we think about the molecular links from genetic variation to complex traits? So as a model, in a couple of papers in the last few years, we proposed what we refer to as the Omnigenic or core gene model. And so as a conceptual framework, we think it's helpful to partition genes into what we refer to as core genes, which have relatively direct effects on a phenotype. So if you think about testosterone, all of those genes in the biosynthetic pathway, we would consider core genes. And then peripheral genes are basically any other gene that are expressed in relevant cell types. So for example, for urate, any gene that's expressed in kidneys is potentially a peripheral gene. And we imagine that their effects flow through regulatory networks. So to illustrate this a different way, we imagine that these core genes are all sitting in much larger gene regulatory networks. And so we have direct effects that flow from the core genes, but actually essentially any gene that's expressed in relevant cell types can have indirect effects on regulation of the core genes, and through that process they can wind up mattering. So here are the key hypotheses that we have. First of all, that most of the heritability is flowing indirectly through weak transregulatory effects. Secondly, that every gene that's expressed in relevant tissues is part of these regulatory networks and thus could have non-zero effects on the trait. Although, of course, probably most of the genes do have small and near zero effects. And then thirdly, following from this, essentially any gene that's a cis-EQTL for some peripheral gene anywhere in the genome might wind up having non-zero effects on the trait. Although, of course, again, most of these are small. So the way I think about this is that you've got some core gene that's got relatively direct effects on a trait, and that's embedded in this larger network. So SNPs that affect the trait you're looking at, the heritability, they may either be cis-EQTLs to the core gene or occasionally missense variants in the core gene, but there's also an enormous number of trans-EQTLs that may have effects. Now, as you know, if you've ever done EQTL mapping, trans-EQTLs generally have very, very small effect sizes compared to cis. They're very difficult to detect. And yet, we know from a variety of different studies that actually most of the variance in gene expression comes from trans effects. So this is a literature survey that we put together a couple of years ago, where we identified a bunch of different papers that use different model systems, different statistical methods, and they tried to estimate how much of the heritability in gene expression is coming from trans effects. And what they find is that, although there's some variation among studies, roughly 70% of the variance in gene expression for typical genes comes through trans effects. So we know now that there's a huge amount of expression variance comes from trans effects, and yet trans associations tend to be very, very weak. And this implies that a typical gene must have enormous numbers of very weak trans regulators. And so we think that actually this helps to explain why so much of the genome contributes to any given complex trait. So if we imagine there are probably tens or hundreds of core genes for any given trait, each core gene may have tens or hundreds of, probably more than that, of trans regulators. This quickly starts to pull in most of the genome as affecting traits. So just to wrap up, what I've suggested is that as a way of understanding why so much of the genome may matter for complex traits, we think it's helpful to make a conceptual distinction between genes with relatively direct effects, which are core genes. But at the same time, we believe that actually most of the heritability is flowing through an enormous number of small trans-EQTLs from peripheral genes that are very difficult to detect, but they're contributing most of the trait variance. And when we want to understand disease biology, really what we're interested in doing is pulling out core genes and master regulators that are sort of that are directly relating genetics to the traits. And I think this is, perhaps one of the big challenges in GWAS system now going forward is to figure out systematically how to do this. But at the same time, for another important goal of complex trait studies is polygenic prediction. And for that, we actually really don't care about this distinction between core genes and peripheral genes very much. What we really care about just is effect sizes. So, if we think about these key challenges, polygenic prediction, as I said, mechanism generally is not very important, but very large sample sizes are essential. For biological insight, I think we really do need to get much better at interpreting GWAS. One thing we've been very interested in my lab, in collaboration with Alex Marson and others, is developing perturbation methods with CRISPR to build networks. I think we need better gene annotations. We need more GWAS of intermediate phenotypes. And then lastly, I just want to thank the key people in the lab who've been involved in this. So Evan and Yang really started this project. Chuan Yao and Yang did a lot of modeling work that was really helpful, at least for me, for thinking about this. And then NASA and Sahin have led the model traits work. So I'll end here. Hand over to, I'm not sure who's leading this, Steve or Dermot, and hand over to discussion.
Well, thank you, Jonathan. There's this kind of brings up so many potential questions of the answers that we're trying to look for in inflammatory bowel disease about all these minor variants and also really the major variants that we have. I wonder if Colleen, if I can show a slide, I guess if I can have control of the share screen.
Yes, you can have control.
Let's see. So I don't know how that, if that's coming up as how it's coming up. And so one thing is in inflammatory bowel disease, we have these major hits, which you can see in this diagram where the width of the height of the variance is proportional to, or the height of the loci given by gene names is proportional to the degree of variance that they,
could you expand that, please, so it's a little bit bigger?
Yeah. Is that better?
Yeah, thank you.
How about that?
Yes, great, thank you.
Okay, so yeah, so here's the major genes for Crohn's disease is NOD2, which in homozygotes of mutation carriers have about a 20-fold risk, and in heterozygotes about a 3-fold risk, and it's dominant in European populations. And this bar is that of European populations. And then we have interleukin-23 receptors that have about 2-fold, actually mainly protective, but there's at least 4 main haplotypes that are associated with risk. And so from your talk, just now concentrating on these European populations, it would seem that maybe what you're saying is that although we know these mechanisms of NOD2 interleukin-23 receptors are both involved with NOD2 as a receptor to bacteria that we believe in the gut and the body's immune reaction to it, and interleukin-23 modifying the cytokine response in the body and degree of inflammation, that a lot of these minor variants may not be directly kind of affecting the immune system are Crohn's disease pathophysiology and unique independent mechanisms, but maybe acting on these major risk variants. Is that kind of what you're proposing?
I think that's plausible. I mean, yeah. So I think it is like you do have a luxury in IBD that a lot of the hits are actually really interpretable compared to compared to many other diseases where I think that's much less the case. So I mean, our experience for, okay, so for those model traits, for example, I mean, we think there's probably a few dozens of hits within the genome-wide significant set that are pretty clearly, directly involved in the biology. As we go down the significant list, it's, like many of the genes, it's less clear to us from prior annotation. Beyond that, I think that, we estimate there's, as I said, on the order of 10,000 variants that that have non-zero effect sizes, and we think that the vast majority of those that are really not very significant, but have non-zero effects are probably highly indirect. So where exactly that cutoff occurs on this list is hard to know in advance. I think what usually happens is that that like the top of the list is highly enriched for genes that are sort of relatively tightly involved in disease processes, and then as you go down the list, the fraction of variants that are relatively peripheral is increasing. I mean, inconsistent effects across populations may in fact, as you said, have perhaps that is an indication that the variants have relatively less direct effects. I see that Carl Anderson has raised his hand.
Yeah.
So I will unmute him so he can ask his question.
Sure.
Hi, Jonathan. How are you doing?
Hi, Carl.
Great talk. I suppose my question was then more of a kind of follow-on to Steve's comment. It does make me nervous that people might be overinterpreting what you mean by the top hits. So I think you're not saying that we should be stopping doing GWAS now that we've got our 240 loci, because we've now got into the realm where we're identifying these non-core peripheral hits. I don't think that's actually what you're suggesting. Am I correct?
Yes, thank you for that point. I think that, I think that the bigger the GWAS gets, the more clarity you get on, what are the core processes that are involved. And, it's, I guess I think as you go down the list, like on average, the variants that are nearer to the top tend to be more important. But as the sample size gets bigger and bigger, you get sort of better sorting of the list, and I think that's extremely helpful. So we did some, I mean, even for the model traits, which are much simpler than any disease, like for that, it's clear to us that if we had smaller sample size, it'd be much more difficult for us to actually pull out the relevant gene sets and make sense of them. So I think that in general, large sample sizes are extremely helpful. I would agree with your point, Carl.
I mean, in the last GWAS we identified something like 25 new hits, and there were genes that lie in pathways that current IBD drugs target. So kind of locus number 215 to 240 is clearly still identifying druggable targets. And so to me, that means that either we're still in the core range and we haven't yet reached the peripheral genes, or if we have, then it means that, well, and I think that's the case. And if we haven't reached the peripheral, then it means that this distinction between core and peripheral perhaps isn't so useful in terms of trying to identify genes that might be identify good drug targets.
Yeah. So okay. So this, I mean, I agree with everything you're saying. I mean, if you think about, the type of data that we're getting from GWAS is it's the association on a SNP, but that's a very indirect measure of how important a gene is, right? Because it depends on, how important is that SNP for, for modifying the function of a nearby gene, and also what's its allele frequency. So the signal strength scales with P1 minus P, and so basically there's a lot of chance that determines that the order of the list. However, as you get a larger sample, at least you get one thing you do get is better estimates of those effect sizes. So you're getting better sorting of the of the true underlying list, and that has to be helpful. But I think it is clear from lots of different traits that there are interesting signals that are enriched and relevant pathways, through hundreds of hits. And and I guess I think of the the top few hundreds of hits are still very high in the hit list if you assume all hits are 10,000.
Yeah, exactly.
So I think some people so some people have taken what I talk about to think that I'm against GWAS. I'm not against GWAS at all. I think it's the best tool that we have for dissecting these questions. I'm a big advocate of it. I think that we have, it's a really challenging problem to take the output from GWAS and extract biology from it. And if it was easy, we would have already done it 10 years ago, and we're getting there.
Indeed.
I see Robert.
Jonathan, just a quick question, just going back to the urate stuff. I thought, first of all, I enjoyed your talk, thank you. And it was fascinating to hear about the urate story. I was trying to remember my urate biochemistry, so it was good to see that put up in front of us, actually. But one thing that I was thinking, as well as understanding urate biochemistry, and therefore using that as a framework to interpret the GWAS findings, we also know quite a lot about some of the things that precipitate gout, and drugs including, and also therapeutic options for gout as well. Did you sort of take this one step further and start to look at how some of those drugs might interact with some of the genetic variants that you've seen, or some of the things in the pathways, either things that precipitate or can be used to treat gout? Because that's what we're after. We're after druggable targets, and you had a framework here to do that potentially.
We have not looked at that. That's actually a great idea. We should look at that. The one thing I do know is that from previous work on urate, the relationship between urate and between the urate variant, the relationship between effective variants on urate and effects on gout is extremely tight. So there's the correlation in effect sizes for the biggest hit variants is close to 1. It's sort of about just about the best example of Mendelian randomization I've ever seen. But looking at the drug effects is a great idea.
Thanks.
I'm wondering if to dissect some of these, we should, in a way, look at endophenotypes. For example, in parallel to the urate, take some of the major enzymes and see if some of these GWAS hits modify that. And the same with Crohn's disease, like look at, for example, tumor necrosis factor, which we don't have an association, but we know biologically is very important to the disease, and look at GWAS association with TNF levels.
So in general, I think I'm a huge advocate of measuring intermediate phenotypes when, at least in settings where you have some ideas, as you guys do, of like what are relevant things to measure. So our experiences with these molecular traits has been that they are so highly interpretable, and also when you do Mendelian randomization with these, you get a bunch of connections between these ones, and for the traits we've been looking at, it's mostly metabolic phenotypes. But I think that is an extremely powerful way of understanding the different biological pathways that are leading to disease. And I think the other thing that the GWAS of these traits does is it, you can come up with a bunch of different, you can come up with different kinds of mechanisms that can affect a trait. So for example, for urate, we thought in advance about the biosynthetic pathway and the transport, but the genetics argues that actually the transport is where virtually all of the action is. In contrast, for testosterone, for female testosterone, it's all in biosynthesis, and male testosterone, it's all in the regulatory pathways. And these, once we'd seen the GWAS, we were able to kind of piece this together out of the literature. Like all of the mechanisms are known about, but we were not able to find anything in the literature. We also talked to endocrinologists. We were not able to find anything that sort of told us in advance this is what we should expect. So I think that actually, even when you know quite a lot about the biology, the GWAS is really illuminating about what's the relative importance of different kinds of processes.
Thanks, Jonathan. Bob Karp has a question.
Hi, Jonathan. That was a wonderful talk. I'd like to come back to urate and questions related to what Dermot asked. The major medication for gout is allopurinol, which in fact inhibits urate biosynthesis. So it's an example of a medication that targets a gene with a small effect size. So and allopurinol works, for which I'm very thankful. But I imagine there are people for whom it doesn't work. So I was just wondering, given that the transporters have such large effect size, have there been efforts? Has industry jumped on some of them as targets?
Thanks for your question, Bob. Those are great questions, and I really don't know. I don't know if anybody knows more about this than I do. That's fascinating. I'll have a look into this after the call.
Okay, and if I may ask a second question, related to IBD. So, we have a large number of hits, varying effect sizes, where we've not yet defined a function. Just in those cases where we don't know the function, apart from the effect size, what other information would we use to make a guess about whether a gene that comes up in GWAS is a central gene or a peripheral gene, or can that only be done through constructing networks?
Well, okay, so there's the various kinds of functional annotation information, things like this that already exist. I believe that this type of information, the sorts of data that we have right now, are not sufficient yet to really unwrap these problems. So one thing that we've been really interested in the lab is developing high throughput ways of inferring regulatory networks, so in principle, we think that probably most of these effects are flowing through trans-EQTLs, and so one would like to measure those, but you just can't get a big enough sample size to do that. So working with Alex Marston, we've been doing some work in primary T cells, where we're building these high throughput CRISPR screening perturbations, and then measuring effects on target genes of interest. There's a whole bunch of different kinds of methods in that space that people are developing, but I think that I'm optimistic that in the next few years, sort of really high throughput perturbation methods and and measurement. So we're using like a flow-seq output, but there's also methods like perturb-seq and crop-seq that are using RNA sequencing as an output. I'm optimistic that those will provide ways of telling us a lot more about regulatory networks inside the relevant cell types, and this may actually be a way forward for understanding these questions. But it's not something we have on hand right now.
Thank you.
Thanks, Bob.
So, Dr. Pritchard, we actually have a few questions in the chat.
Okay.
So the first question is from Paul O'Reilly. The question is, I think Omnigenic Model predicts extremely large associations between the expression of core genes and outcome. Where are we now in terms of evidence for that from gene expression data across different traits?
Great, thanks. So I think this is a really interesting question. So I think one of the mysteries in GWAS right now is this observation that a number of groups have made. We've been puzzling over this for a while, which is that the links between EQTLs and disease are not as striking as one would expect, given our sort of general conceptualization that non-coding variants are affecting genes through cis-EQTLs. And so there's in particular, there's a couple of methods or several methods like TWAS and PredictScan that are aiming to do exactly what you're talking about here, where you can sort of build these predictive models of the links between SNP variation and gene expression, and then ask whether those are predictive of disease. And when people run these kinds of models, they get a bunch of hits, but they tend not to be very interpretable. And I think it's not really been as clear a win as I expected that it would be. So I think it's not entirely clear what is sort of driving this gap between cis-EQTLs that we measure and disease hits. My current sense is that actually there's something complicated going on, that the disease hits are often more tissue or cell type specific or condition specific than many EQTLs that we measure. EQTLs are more clustered around transcription start sites than GWAS variants. And so I think that there's a little bit of a gap to it right now between what we measure in EQTL studies and what GWAS variants are picking up. This is sort of an important gap to fill. So that was sort of a long answer to your question, I think, but I think this is sort of a gap in our understanding right now that you're highlighting.
All right. We have another question from Alex Forrest-Hay. What are your thoughts about the concept of metabolic risk scores as larger and larger data sets become available looking at lipids, metabolites, and proteins, etc.? We are seeing some sealer risk profiles for complex common diseases.
I think that these kinds of polygenic scores can be really helpful, and we've also been interested in methods where we compute polygenic scores on on various different metabolites and then sort of put them together to as independent predictors in prediction of risk for metabolic syndrome kinds of traits. So yeah, I'm not sure if I have much to say beyond that, but I think I'm bullish on this kind of approach.
No problem. I see that Dr. Rue has his hand raised. Maybe he can answer his question in the last 2 minutes. So yeah.
Okay, yeah, that'd be great.
Too many questions. It was great talk. Thanks so much for that, Jonathan. I think, you know, what's hard for us to discuss right now is this difference between core and peripheral genes, only because, you know, you haven't really defined for us what those peripheral.
Oh, sorry, you've gone quiet. I lost you for a moment there, Jonathan.
I'm back. Yeah, I don't know why I ended up going on mute, but you know, not knowing what your definition of these peripheral variants is kind of hard to sort of have a discussion on how you define them, how to interpret them. But, you know, you have to imagine that there's not a, you know, a clear, you know, cliff where you go from one to the other. This is, you know, biology. So there's a continuum. So you have to imagine that the further you go out, the less specific to the phenotype you're actually interested in. And so have you given much thought about how to combine multiple different phenotypes in your analyses to try and get more specificity if you go for a broad range of different phenotypes, unrelated phenotypes?
Well, so to your first point, I mean, I guess I largely agree. We're trying to, you know, come up with some conceptual model for thinking about the problem that's obviously
which is great. You have to, right?
Yeah, that's right. And you know, I tend to think that there's so I do think there's something fundamental about cis versus trans EQTLs. So so we know that trans EQTLs individually tend to have very small effect sizes, and so part of what I wanted to do in the way we we actually sort of shifted our definition of core genes slightly between the Boyle et al. Paper in 2017 and the Liu et al. Paper in 2019. So in the second paper, what we the way we defined core genes is that they have a direct causal effect that's not flowing through regulation of other genes. And so the reason for that definition is that it means that variants that are in cis to the core genes can have effects as cis EQTLs, but variants that are in cis to anything else, their effects have to flow as trans EQTLs through the network. So I think that there is, so there's definitely edge cases where that definition breaks down a little bit. But anyway, I think that, yeah, I think that there's some utility to, you know, to trying to divide genes in this way, even though I agree with your larger point that it's there's, you know, we're kind of flattening out a complicated space into something that's got, you know, that's binary. So your second question was about sort of combining different phenotypes.
You know, if you combine, you know, schizophrenia with, you know, type 1 diabetes with, I don't know, height, you know, you should at some point see that, you know, a definition of core versus peripheral starting to emerge based on, you know, specificity to a phenotype or to related phenotypes. Because to some extent, if you're getting to 100,000 variants, you know, for some traits, it's hard to imagine that those variants are, you know, specific to that one trait. They have to be involved in other traits as well. So, you know, to get some sort of like, I don't know if it's a score, a specificity score over a broad range, you know, when you're looking at these very, very large data sets that have so many different phenotypes, obviously it's going to be hard to, you know, define independent phenotypes, but there might be a way to sort of, to sort of come up with a score on the phenotype in terms of their relatedness. Obviously, we make mistakes when we do that, but that might be a way to also just sort of try and figure out which ones are specific variants.
That's interesting. I'd have to think a little bit about how to implement that idea. But I mean, one thing we have thought about is like sort of how to interpret genetic correlation. So in the context of our model, basically when you have genetic correlation between two traits, what that implies is that there's either overlap of core genes or at the minimum that the core genes have correlated, like they tend to be in the same path. We have shared core genes, and basically anything that's a trans EQTL for one, so trans-EQTL for a core gene for trait 2, and so basically that's sort of an interpretation of genetic correlation in that model. Whereas two traits that are mediated through the same tissue, but the core genes are completely unrelated,
Right.
then there what you get is like, there are predictions that you'd have variants that are shared, but there's no correlation in the direction. So the genetic correlation is basically 0, but as you said, you wind up getting overlap of variants just because there's only a finite number of regulatory variants in the genome. So I think there's more interesting things to be done in that space.
Looking forward to watching this in the few years to come.
Yeah.
Jonathan, thank you. That was absolutely superb. And as you can see, it generated the interest that we hoped. And we could actually have continued going on, but I think I have the difficult job of stopping the interest and releasing you as such. So thank you. We really enjoyed that. For everyone who tuned in, thank you very much. And thank you for your interest. The next one of these is on August the 6th, at the same time when Thad Steppenbach will be joining us, talking about intestinal epithelial defect triggered by genetics and environment as a target for IBD therapy. So please join us for that. Thanks again, Jonathan.
Thanks, Jonathan.
Thank you so much, everybody.
Take
Great to get to talk to you, and thank you for your questions and comments. Thank you, everybody.
Thanks, Jonathan.